Welcome to Highlighting Bladder Cancer Clinical Trials, Muscle-Invasive Bladder Cancer. This is a patient insight webinar from the Bladder Cancer Advocacy Network. We'd like to thank our sponsors, EMD Serono, Ferring, Insight, Genentech, and Merck for their support of the Patient Insight webinar series. Dr. Peter Black is a urologic oncologist at Vancouver General Hospital, a research scientist at the Vancouver Prostate Center, and a professor in the Department of Urologic Sciences at the University of British Columbia in Canada. Dr. Parminder Singh is a senior associate consultant at the Mayo Clinic in Arizona in medical oncology. He's also a member of the Southwest Oncology Group GU Committee, playing a critical role in designing clinical trials in bladder cancer. And Rick Bangs is a bladder cancer patient and a prostate cancer patient survivor, and he's worked as a patient advocate in a variety of roles, including research advocacy, government lobbying, educational support, support groups, one-on-one support, and fundraising, and he's been a longtime friend of Beacon.
So the topic today is muscle-invasive bladder cancer. And we want to focus on what's going on in the field of clinical trials. I'm going to start with a bit of background just on how we treat muscle-invasive bladder cancer. So when we're talking about muscle-invasive bladder cancer, of course, we're distinguishing that from non-muscle invasive bladder cancer, which was addressed in the last webinar on this clinical trials series. And so here, as the diagram shows, these are tumors that are going into the muscle or beyond. And these are tumors that we typically treat with removal of the bladder. That's the mainstay of treatment. That's the radical cystectomy. And at the time of cystectomy, we will also remove lymph nodes, the pelvic lymph node dissection. And of course, we have to replace the bladder with a segment of bowel, whether it's a neobladder, an ileal conduit, or something else like an Indiana pouch. So it's really a three-part surgery. This is sometimes preceded by chemotherapy, cisplatin-based chemotherapy, which we call neoadjuvant chemotherapy, or it can be followed by chemotherapy. So if a patient goes straight to surgery and has a significant cancer at the time of surgery, we'll sometimes give chemotherapy afterwards. And some patients we will treat primarily with radiation, usually in combination with chemotherapy if the patient is eligible to get chemotherapy. We sometimes refer to this as trimodal therapy because it includes a transurethral resection, which is the initial biopsy of the tumor, followed by the radiation given together with chemotherapy. And this is something that Parminder is going to focus on later. Typically, we would reserve this for tumors that are maybe a little bit smaller and the patient has good bladder function. It's just a solitary tumor. And so there are some specific criteria we use to select these patients. So if we look at this, these general treatment domains for muscle-invasive bladder cancer, it can highlight where the unmet needs are, and we try to design trials, clinical trials, to address unmet needs. So with radical cystectomy, one of the big things is that it is associated with a lot of complications. It's major surgery, and we would like to do what we can to reduce complications, to enhance recovery and improve outcomes. And so I'll come back to a trial that is going on that addresses this issue. Another important surgical question is how extensive should a lymph node dissection be at the time of radical cystectomy? Do we just remove a little bit of tissue or do we remove a lot of tissue? There's a feeling in the field that more is better and that we should do an extensive node dissection, more up to a point, of course. But there's a risk that it could just increase complications without being beneficial. And in other cancer sites, trials have shown that it's not necessarily beneficial. So this is another important question that we can ask with a clinical trial. With respect to chemotherapy, we know that chemotherapy causes side effects. We know that patients generally aren't excited about receiving chemotherapy, and we'd really only like to give it to patients who we expect will have a good response to chemotherapy. And the best way to potentially identify patients who will benefit is with biomarkers, especially molecular biomarkers. And so there's a lot of work ongoing right now to develop biomarkers that will answer these important questions. And so if we can identify who's likely to respond, we would give those patients chemotherapy. Other patients would go straight on to surgery or radiation and would be spared both the side effects of the chemo and the potential delay in the surgery. So the first trial I would like to highlight fits in that first question of how can we improve outcomes and reduce complications and ultimately improve quality of life around the time of radical cystectomy. And this is a Swog trial, so it's a cooperative group trial. It's all these trials have numbers, so this is 1600. And this is a trial that's looking at a special nutritional shake that's to be taken before surgery for several days and after surgery. And it has a special content, so special fatty acids and amino acids and other trace elements that are thought to enhance immune function and decrease the stress of surgery and improve outcomes would be the hope. And so in this trial, the special immune modulating drink is going to be compared to a placebo drink. And the main outcome that's going to be measured in the trial is the rate of postoperative complications after cystectomy. They'll also, the investigators who are listed at the bottom there, Jill Hamilton-Reeves and Jeff Holzbeierlein are the two principal investigators. They will also be testing whether the immune modulating drink can reduce the rate of infections after surgery, muscle wasting. So a lot of patients will experience not only weight loss, but actual muscle wasting after surgery. The rate of high-grade complications, so not just minor complications, but the more significant ones. The rate of readmission to the hospital. There'll be quality of life questionnaires, and they'll even look at survival. So does something as simple as a special nutritional shake around the time of surgery actually enhance survival outcomes? There'll also be some interesting biological tests that'll be done with blood, urine, and tissue in patients on this trial, where the investigators will look at actually how the immune system is responding to the drink versus the placebo, also the metabolic response and the overall stress response to surgery. And so this is going to be a trial of 200 patients. Patients will be randomly assigned to one of two arms. So there is a very simple trial design where one arm will get the shake, the other arm will get the placebo drink. It starts 5 days before surgery, it continues for 5 days after surgery, and all these different endpoints are captured. Blood, urine, and tissue, as I mentioned, is investigated, and I think there's also a bone density study after 30 days. So this is a trial that is launching anytime now, and will accrue across the US and Canada. So this slide here, SWOG S1011, this is a trial that has actually completed accrual, so it's no longer open, but we're waiting for the results as more follow-up information is gathered on the patients. This is a trial that is looking at the extent of lymph node dissection, trying to determine what is optimal at the time of radical prostatectomy. We know that, or we think, if bladder cancer has spread to the lymph nodes, that it's beneficial to remove it, and we can still cure patients with node dissection. And so if we remove more, we might be able to cure more is the basic idea. And so this is a trial that includes any patient with non-metastatic muscle-invasive bladder cancer who's fit for radical cystectomy. They can or cannot have neoadjuvant chemotherapy, whatever the treating physician is able or wants to do. And then they're randomized to either a extended or a standard node dissection. You can see, based on the anatomy here, that the standard is just down in the pelvis. we talk about where the artery split. So this would be the split of the iliac artery into an external and an internal. So we would define the standard lymph node dissection as something below that towards the bladder. And the extended dissection then goes all the way up to this is the aorta here where it splits. So a lymph node dissection is really just a removal of fatty tissue around the blood vessels. And the pathologist subsequently actually sifts through the tissue and identifies the lymph nodes. And so if we go a little bit higher and remove a little bit more, it could have a positive impact, or it could have no impact and increased risk of complications, as I mentioned. So this is a key trial that has already enrolled approximately 650 patients. And the outcome that the trial is measuring is patient survival, disease-free survival after several years. The diagram indicates there that patients can also have adjuvant chemotherapy after surgery if they need to. So since it's looking at I think it was a 5-year endpoint, it takes several years of follow-up before the trial is actually reported back to the scientific community and as well as back to patients. So there we are. Sorry, it's 3-year disease-free survival. So then another trial in the area of biomarkers is this trial that looks a little bit complicated. This is one that Tom Flagg in Colorado and Dan Theodescu, who was also in Colorado, is now at Cedars-Sinai. They've led this trial. And this is a trial where patients are really being treated according to the standard of care. So they have a TURBT to remove the bladder tumor. They are found to have muscle-invasive bladder cancer, no metastases, and they have to be eligible for chemotherapy. And then they're randomized to 2 different chemotherapies, although the trial is really not to compare the different chemotherapies. I just want to include both of them. I'll come back to that. The patient then has surgery to remove the bladder, and there's then tissue available from that. There's tissue available from the original TURBT and again at the time of cystectomy. And we can measure the response to the chemotherapy. Is it tumor completely gone, or is there still tumor left? And this response is going to be correlated to a biomarker that's called a coxin biomarker, which is based on RNA expression in the tumor prior to chemotherapy. It's a special algorithm that uses this RNA data to predict whether the patient is likely to respond or not. And that has been tested in other small series of patients, but this is now the prospective randomized trial to see if we can validate it in this trial. So at all time points, so before chemo and after chemo, we'll have tissue, blood, and urine collected. We'll do all sorts of different tests, not just the coxin model, but primarily the trial is designed to validate the coxin model to see if it really does predict pathologic stage. And then there's also this discovery component where new markers can be tested as well, and not just the coxin model. And since the endpoint of this trial is the response to chemotherapy, which we really know at the time of cystectomy, this is not going to require long-term follow-up. And so this trial also just finished accrual earlier this year, and the molecular analysis is ongoing, and we may have initial results before the end of this year, where we'll see if the coxin model adequately predicts outcome. Again, as I highlighted earlier, if it did predict outcome efficiently, then we could spare chemotherapy in the patients who are predicted not to respond, and we would prioritize chemo in those predicted to respond. So another very important trial.